Good evening, everyone. But anyway, a warm welcome to everybody here this evening and to those online. So thank you for joining us. We're going to have a very exciting Understanding Health talk, which is a collaboration between Bournemouth University and University Hospitals Dorset. But before I start that, can I just say to you that the facilities, the toilet facilities on this side are the male and non-gender, you know what I mean. And on this side, almost at the door, are the female. I know that because I've just been there. So if the fire alarm goes off, there is not a test expected. So please, can we go in an orderly fashion out of the door, and we follow that gentleman there in the check shirt, and he will lead us to safety. Anyway, having got that out of the way, University Hospitals Dorset and Bournemouth University have been working together for a good number of years, all the time that I've been involved with them. 3 years ago, they made that a formal agreement, which has benefited students, it benefited patients, staff, and the community. So it's a wonderful union that we're developing as we go along. You might wonder who I am. I do sometimes. Anyway, my name is Sandy Wilson, and I am a public governor with University Hospitals Dorset. And what I'd like to do is just steal a few of your minutes, please, before the talk starts, just to talk about membership of University Hospitals Dorset. We're a foundation trust, and therefore there is a requirement for us to have members, but it's a very positive thing, because as members, you get informed of things that are happening, like our talks today. You get informed of things that are happening. And also, if you wish to become involved, you can have the opportunity to be able to feedback on things that are taking place, or the ideas or comments that you may have at the hospital. We're trying to provide those opportunities through a variety of channels for people to be able to contribute. And certainly the hospital at the trust at the moment is going through a fantastic transformation. We have secured over £500 million, and that is helping to improve the services that we can provide to our community, and alongside that with Bournemouth University. So I urge you all in the room to perhaps pick up one of our leaflets, or there is a QR code on our display. And for those of you who are online or even in the room, you can pop onto the UHD website, and then if you click on About Us and then Supporting Our Trust and then Becoming a Member, the formal things that you need to do will be very, very clear, and you can become a member. It's totally free, and you can choose what level you'd like to be involved. So that's my little plug this evening on behalf of the trust. But now it's my pleasure to introduce our two fantastic speakers. And they're going to unravel for us the mysteries of modern pathology and how they interact with clinical services to provide diagnostic support, and how pathology laboratories are making a more personalised approach to healthcare. And no doubt will include how our new state-of-the-art pathology unit that we've got will play its part. So it's my pleasure to introduce these two wonderful speakers. So we have Nathan Bourne, who has a doctorate in structural biology and is a fellow of the Institute of Biomedical Science, and is the biomedical scientist who is our governance and quality manager at UHD. And I've tried to practice this name, but not very well. So we have and she's been very kind, Dr. Mantzouratou, and I've got it wrong again, who's once again a fellow of the Institute of Biomedical Science, a fellow of Higher Academic Education Academy, and a principal academic in human genetics. Thank you, and over to you two.
Hi everyone, it's lovely to be here. I got asked to come and talk about pathology, which is something I love to do. Pathology is an amazing place, and it kind of goes under the radar a lot about what we actually do in pathology. So for the next 20 minutes or so, I'll be looking at a lot of what pathology actually does. It's not just a big building that blood tests go in and results come out, but there's a lot more into it as well. And as we go through, you'll see what all the different sections of pathology are, and also what some of the newer developments that are coming in as well. So this is pathology over at Bournemouth. We've just built a brand new hub. This is a brand new Dorset Pathology hub. It's purpose built. We just had the opening a couple of weeks ago. Now this building is the state of the art. We've designed it to future-proof pathology in the future in Dorset. This is going to be the centralised hub where a lot of the testing in Dorset will come to. It's a nice, lovely building there. You can see Bournemouth Hospital there in the background. But this is some of the things that we do. So we process about 9 million tests a year. It's quite a lot. 70% of all conditions that are diagnosed in the NHS come through pathology. It's open 24 hours a day. We have about 200 staff manning that, and it's a green building as well. And so if you've managed to go on the top, there's lots of little solar panels on there and everything. It's a lovely building, a lovely place to work. But it's not just a building. There are several things inside of it as well. And it helps with us to diagnose and serve the population. If anyone comes into a hospital or to a GP, they have the bloods taken. Those bloods will come to pathology. Everything kind of comes through pathology, and nearly everyone who comes into the hospital or to a GP will come through pathology. Now, it's not just a building. Pathology is separated into separate areas, into 3 separate areas. We have blood sciences, cellular pathology, and microbiology. Blood sciences is what it says, really. It's looking at bloods. It's looking at all the different kind of sections. According to that, we have biochemistry, blood transfusion, coagulation, hematology, immunology, and molecular pathology. Cellular pathology is looking at cells, as the name would indicate. It looks at 3 types of cells in our local labs. We've got histology, which is looking at tissues, cytology, which is looking at cells within a fluid, and then andrology, which looks at male reproduction as well, so it looks at sperm. And then finally, we have microbiology as well. Microbiology is where I trained in, and this is the lab that I know the most. That's why there's the most slides about it in this actual presentation. And so this is looking at infection. In my time in microbiology, tested absolutely everything, and to look for all kinds of infections out there. So you look for bacteria, for fungus, for parasites, for viruses. It's all kind of in there, and we'll go into a bit more later on. So we're going to look at just a few of the different sections, a few of the different labs, and what they actually do, and how that will impact the health. The first one and the main one is biochemistry. These are our biochemistry analyzers in the hub. These are brand new. We do around about 5,000 tests a day on these. I call biochemistry the real workhorse of pathology. These are some of the test profiles that we have. We look at renal, liver, you look at your bone, lipids, endocrine disorders, therapeutic drugs, toxicology, proteins. There's lots of different things that biochemistry does. But what we do all these tests, and we do them it takes around about an hour to do. Biochemistry is one of the things that most people have. They take the blood, it will come in, and they will look at your blood biochemistry, and that's an early indicator to see if you're okay or not. If your levels are all fine, then the person will get released from the emergency department or from the GP. The GP will say you're fine. If not, then it will require further investigation, and then we can go further into what is actually wrong. So biochemistry is one of the first ports of call for anybody when it comes to investigations. Probably one of our most important ones is blood transfusion. in Bournemouth as one of the major acute services, we have to have lots of blood available to be able to transfuse people. We have a rule, so it's the two sample rule. So that when people come in, we have a blood group, but we have to do that twice to make sure that we get the right blood into the right patient. So this is our lovely machine, the IH500. It's a brand new machine. Actually, it's about a year old actually, but it's almost brand new. And this will test out for blood groups. I'm a blood group donor. I'm an A negative, and so when I give my blood, and when I donate my blood, I know that it goes to certain places because I know that it's very needed. And this is one of the most important places that we have in the hospital. So these are some of the things that we have on the Bournemouth site. There's been a lot of work there. You can see a big building coming up at the front called the Beach Building. This is going to be one of the main areas for people to come in, and it's going to be needed for blood transfusions. So people in critical care, people who are coming in for emergency departments, but also it's a major cancer centre as well. We have a haematology, or the haematology wards are over at Bournemouth as well. And so there's a great supply, there's great demand for bloods for people who come in. So these are some of the things that we look for, that we use blood transfusion and to transfuse blood into people. So potential bleeds, so people who are bleeding out, pre-operative. So again, if we have theatres on site, then we need to have blood available then. People who have anemia, people with leukemia or cancer patients, these all need blood, and our blood transfusion laboratory makes sure that the right blood goes into the right person, because if it goes into the wrong person, then it could end up really, really badly. So that's one of the most important things that we have to do. So we do around about 25,000 to 30,000 tests a year for that. So as I said, a lot of that we get done twice, because we've got to make sure that when we do our blood grouping, that is done so many times to make sure we got the right blood for the right patient. Right, linked into that is haematology. Haematology, if you ever had your bloods taken, it's the little purple top that we have. So this looks at basically the blood. It looks at the red cells, looks at white cells, looks at platelets. It looks at all the different shapes and all the different sizes, looks at how old they are, it looks at how many you have, and each one of them might indicate a different thing. For example, someone with white cells, if someone has lots of white cells, it might indicate an infection. If someone doesn't have a lot of white cells, it may indicate some kind of leukemia maybe. If there's a different cell type, it may indicate something else. There's lots of different ways that we use haematology to try and predict what's going to happen and what's wrong with you. Platelets is another thing. So platelets is used for coagulation. So if you cut yourself, it's the platelets that form the scab on there. If you don't have enough platelets, then you might not be able to clot. So then you might need to get some anticoagulants like warfarin or something like that. So haematology is, again, with biochemistry, it's one of the major things that we have to be able to accurately diagnose patient when he comes in, when they come into pathology, come into the hospital or to a GP. We have an immunology department. Now, we at Bournemouth in the hub, we have our immunology department is the only one in Dorset, so everything comes through there. And generally they do two things. They look for allergies and they look for autoimmune diseases. Now, my son is allergic to peanuts. What happened is that they took his blood, they sent him to the immunology department, and they had a little bit of the peanut allergen, and they did a test, and it came up that he was positive for an allergy to peanuts, which is a shame because I really, really like peanut butter, and I can't eat it around him anymore, which is a bit of a shame, but that's okay because he's all right. We also look for autoimmune diseases as well. So things like rheumatoid arthritis, lupus, celiac disease. All these are diagnosed in our immunology department. So I know celiac is quite, well, not popular, but it's something that is quite prevalent at the minute. It's a lot of gluten-free. If anyone is a celiac, that has been diagnosed in our immunology lab. Oop, skipped one there. We also have a molecular pathology lab. So this kind of gets split into 3 sections. We have flow cytometry, molecular, and research. So flow cytometry looks at white cells, and so it looks at the different types of white cells. It looks at how old the samples are. It looks at the leukemia. It looks at lymphoma. It looks at little markers that's on the white cells. It also does some counts as well. So, for example, CD34 is a white cell that is produced when you do stem cell harvests. And so when someone has leukemia, we do stem cell harvest to help produce this CD34 cell. We do the counts to see if the stem cell treatment is working for the leukemia. It's very similar for the HIV patients as well. So CD4 is a T cell, and it's an indicator if someone has a HIV infection. If someone's CD4 count is low, that means the infection is going ahead. But if the CD4 count is high, it means the treatment is working and someone can carry on with their daily lives. We also have a molecular path, which I think is incredibly interesting. So this looks at the actual DNA of cancers. So we're looking at, we can look at the molecular structure and also the DNA sequences of particular cells and particular cancers. And with that, we can do specific treatments. If a particular sequence is there, they can have a specific treatment. And we can track that as we go through the treatments and go through the patient journey. Because if that particular sequence is gone, it means the cancer's gone as well. And so it's very interesting of how it all kind of goes along. We also do our own research as well in companionship with our research department at Bournemouth and Paul as well. And so we do a lot of clinical trials, and so we do a lot of clinical trials and areas with the research department, and to be able to go through those clinical trials. We have microbiology, and I know a bit more about microbiology, so I might be able to tell a little bit more about that. So we look at, and if you can be infected with something, we will test it. I've tested absolutely every kind of sample you can imagine. The main thing is really we've got to find out what the infection is, and then provide options to how to treat it. And so the consultant, we liaise quite a lot with the consultants, and they will talk to lots of different people and their own clinicians as well, the patient's clinicians, and provide advice about how to treat it. So one thing that is quite prevalent is sepsis. So in our new hospital, our new pathology department, we have a new blood culture machine. We had previous one, but this is just a bit of an upgrade. And so what we do is we look for sepsis with it. So a patient will come in, they will have a blood test, and they will take some blood and put it into blood culture bottles. Now these bottles have a little sensor at the bottom. It detects a respiration. So if there's something in that bottle that is respiring, either oxygen levels are going down, carbon dioxide levels are going up, the sensor will detect that and be able to tell you there is something growing in there. Now the biomedical scientist will be able, well, the machine will go beep, 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 and detect this, and alert the biomedical scientist to say that there is something that is growing in this particular bottle. We need to identify it. So the biomedical scientist will take the bottle and put a couple of drops on an agar plate. This is an example here. This is probably Staph aureus from what it looks like. And we'll investigate, find out exactly what it is that's causing the bacteremia. And this is one thing that's kind of vital. And with sepsis, it's got to be very, very quick. So you've got to be able to identify what the markers are. This is where we link in with a lot of the other labs as well that will have indicators for sepsis. In biochemistry, you might have a look for things like CRP and ESR to be able to find out if there's an infection. But with this, following this, we need to be able to identify the bacteria and then be able to treat it as well. So once we've identified the bacteria, we do a sensitivity testing. And this is just basically finding out what antibiotic is suitable for us. So there's a number of different tests that we can use. A main one that we use is disc diffusion. It's just little discs with the antibiotic in. And we put it on an agar plate and swarm it with the bacteria. And as you can see here, there's some that's kind of grown up completely, and with it, you see the culture has grown up to the disc. That means it's resistant. But where there's a very wide area where the bacteria hasn't grown, which means it's sensitive. This means that is an appropriate antibiotic to give to that particular patient. We also do molecular testing as well. So when I first started as a biomedical scientist, I started in 2008. It used to take about 2 days to get a flu result. What we would have to do is get the sample and incubate it in with a load of cells and see if the flu virus was there. It would infect those cells, and we would produce a fluorescence there with a certain test kit. Now we can do it in about an hour. And so these are two of my favourite machines, particularly the one on the right. This is the BioFire. Oh, I didn't put the name of it. That's the BioFire over on the left-hand side. That's my favorite machine because you can do about 30 tests and in around about an hour, and it's amazing how it all comes out. It's called a microarray. I'm not going to get into the science because I'm a bit of a geek, but we can look at specific areas that can help people, like a respiratory panels. We can find out straight away within an hour if it's flu, but then if it's not flu, then there's something else. We test about 5 different coronaviruses, a whooping cough, as I understand there's more whooping coughs around. This will test anything that will kind of infect the respiratory system without about an hour. And the same with meningitis and encephalitis. It's a different screening panel. But then if someone is suffering with meningitis, again, it's a very timely thing that we need to find straight away. Do they have meningitis? It can be a very quick disease and very quick progressing disease. To have this test to be able to find out in an hour or so whether there is something there or not is a really, really fantastic achievement. So just moving on to histology. Histology is looking at tissues, and so this is generally we're trying to find cancer, and so this is adenocarcinoma there for the picture. So we get all types of different samples come in, and we process them and we put them onto a nice little slide, and we look at the slide and we do different stains and we have a look for different markers to see if cancer is there or not. This is the basis of histology. Now, one of our new developments with the new hub is that we have digital pathology. So what this is basically a big scanner. So this will create a digital image of each of the slides, and so because of that image has been digitized, that can be sent absolutely anywhere. So it could be sent to New York, it could be sent to Tokyo, it could be absolutely anywhere. One of my consultants jokingly said to me that he's going to retire to Tahiti, and then all the slides can be sent to him there. And then following that from there, because all these slides are now digitized as very high resolution slides, we can start developing artificial intelligence to find out where the markers are. So we use it for second opinions, for real-time digital consultations. Generally, if something is just on a slide, then you know, we've got one or two people will have a look at it, but if it's a digital slide, we can be put on a big screen like this and anyone can see it. We can use it for MDTs, multidisciplinary teams. We can use it for teachings. We can also, as I said before, we can send it anywhere. People can, the pathologist can do this at home or in any location that they want, really. But then we also we can develop the artificial neural networks as well. And so this is developing AI. This is using the tools that we have currently to be able to create all the different algorithms, so we can train it to be able to spot all the different markers and to be able to say, okay, there it is. So this is going to save so much time. If you think about, so a lot of the weight, so if anyone has had a cancer diagnosis or anything like that, the thing is that is the weight of not knowing, and they've put a sample in and they don't know because it's been such a time. So using the AI is going to cut the knowing time down to be able to find out that diagnosis a lot quicker. So there's some of the benefits of using neural networks in histology. So we've got virtual reporting. It's absolutely fantastic. Improvements to turnaround times, we don't need to stain it less. And so those are some of the benefits there. And so this is just an example here. And so of breast cancer pathology. So my mum's got breast cancer at the minute, and hers is it's an estrogen receptor type of breast cancer. And so there are lots of different other types of receptors here. And so this is an example of all the different AI, all the different receptors and all the different markers that you would find for breast cancer. And the AI algorithm has been put in and to have a look at and identify each one of them particular receptors. So it finds out that an ER receptor or a progesterone receptor, it looks at the HER2 status as a particular. If it's positive for HER2, then it can have Herceptin, which is a particular treatment. It looks at the proliferation. Is the cell multiplying? Things like that. So it's really remarkable the things that it can do. And it can also develop heat maps as well. And so this is just some of the things that you kind of see there. So all the different kind of cancers in there. It's still very early in the very early stages, but it's something that we're going to be developing very soon. And this is one of the things that is coming soon. So that's pretty much the end for me. I just got one last slide. So I mentioned right at the start we have a brand new building, the Pathology Hub, based on the Bournemouth site. Now we had a big party a couple of weeks ago, and we invited the first patient who was tested in our labs. His name's Stephen Horns, and you'll see a video of him next. And so his blood was the first patient to be tested, and so we invited him along to cut the ribbon and officially open it. So it's just we asked him afterwards about what we thought of pathology. About the hub as well. So these are some of his thoughts.
It's been particularly impressive. Things were huge impact.
Oh, is this on? Hello? Hello, can you hear me? There we go. Isn't that wonderful? With that, with our opening last week, the week before, I was watching Steve there. He was the first patient, and he just had just the biggest smile on his face all day. And when he came in, I was privileged enough to take him around the pathology hub to show him all the different machines and show him all the different areas. And he was just so appreciative of the fact that we have this magnificent place to be able to diagnose and treat diseases like his. And I'm sure you can saw his gratefulness there in the video, and that is why we do it. We're there to treat patients. We're there. My old boss used to say every morning, "We're here to save lives. We're saving lives." And this is something that I've tried to take into with me as well. Is that at every sample there's a patient on the end of it, and we. Whether it's a negative result or a positive result, that's helping them in their patient journey. And I'm going to pass over to Anna now, who's going to talk a bit more about personalised stuff.
Thank you, Nathan. Just gonna. Hello, everybody. My name is Anna Mantzouratou. I'm a geneticist, and I will talk to you a little bit about something more specialized in the terms of pathology. So, sorry. Thank you, Nathan, for this brilliant introduction to pathology. I do a lot of human genetics, but also lately we're doing a lot of things with antibiotic resistance. But I'm going to talk to you about how we're starting to personalize things by combining both the knowledge that all other sciences give us, but also whether the knowledge from genetics from our own DNA is getting. So this is the front sort of screen in the London in the MS. I think it's a medical the BMA, the British Medical Association, or the Wellcome Trust, sorry, which is in the same sort of area. So there's no one quite like you, and that is really true, because really there's no one quite like you. You're all unique. We are all unique, even if we are twins with somebody. As we grow up, we become very, very unique because we are also shaped by our environment. So when I was passing the Wellcome Trust, I was working around the area back a few years ago. I saw this in the front, and I thought that that was a really nice. So I took this picture quite a long time ago, but my main research is about genetics. I do a lot of reproductive genetics, but also nowadays work with colleagues in university hospitals, all set to actually do research on antimicrobial resistance and how we, from our environment, we become more aware of how microbes change and how they affect our life, but also how we use our genetic information to prognose, diagnose, and manage diseases. So I will make a little bit of introduction about my science and how we use it to help people and a lot of other, in a lot of different areas of life. So, but first of all, a little bit of some trick questions here. So if you think about what comes into your mind when you hear the word genetics, obviously you hear about genes, not the blue genes, but actually our genes in our DNA. You also, maybe, you know, when you were young, you were exposed to Teenage Mutant Turtles and Star Wars, the Attack of the Clones, which is also related to genetics. Recently, though, you have this scientist that in China that he proposed to, he actually did human cloning illegally, and genetic engineering, obviously illegally. He was in jail for a long time. He tried to, he actually not tried to, he genetically engineered 2 embryos to create resistance to HIV. So because the father was HIV infected, and he thought it would be a good idea to try it illegally. So he didn't have permission. The scientific community was very much against it. He presented his findings to very openly to a research symposium, and then afterwards he was arrested, basically. So, you know, so there are genetically engineered humans, you know, walking amongst us. These 2 little girls were born, changed, but the changes that he instigated were not controlled, you know, because technically we know the theory, but technically we are not achieving what we're supposed to be with great precision. So we're not there yet, you know, to genetically engineer humans. But these 2 little girls, we don't know how they would develop or what diseases or what happened, because it was very much an ad hoc sort of situation that this person did. But there are genetically engineering humans working amongst us. So looking at genetics, you know, there is quite a lot of development happening here. But should we be afraid? If we look back on 1997, you can see a mobile phone that I used to have. But also there is this movie came out in 1997. Anyone knows what this movie is called? A lot of my students don't know. A prize for Nathan? The movie was called Gattaca, and it was about a person who, you know, the people were selected to do their own professions according to their genetics, and they were genetically engineering sometimes humans, because if they were testing the embryos and they found some flaws, they corrected those flaws basically, and they were becoming, you know, astronauts and doctors and everything. But if you were not engineered, you were in a sort of lesser class, and you were doing jobs that, you know, do not require a lot of effort. So it was about a human that came from that lesser sort of class as they had it, and sort of fooled the system to become an astronaut, and, you know, was supposed to be so it was showing all this futuristic. Back then it was futuristic, it was like not done yet, but then now everything that's in that movie is actually theoretically possible and has become reality. So here I'm just going to tell you about how unique we all are. So for example, how we're reacting differently to drugs. Why are we digest different foods differently? All this, you have to look to genetics and how we you know, we were healthy, we have cholesterol, different levels of cholesterol even. If it's harder even to beat addiction or be addicted to certain these partly are dictated by genetics, not all the time, but partly. And so just a little bit of background about what we are made of, what is our instructions of life. So inside the Wellcome Trust Institute, so you had the screen before in London. It's just across Euston Station. So they have the genetics exhibition. So inside you have the genomes exhibition. So this is I'm just going to put the pointer. So here we have all our chromosomes. So we have 46 chromosomes, we have 23 pairs, and all of them coming, we have one chromosome from our father, one from our mother. So basically they pair up inside us and they create our unique blend of people. So what's in the chromosomes, basically, inside those genomes exhibition in the Wellcome Trust Institute, you have the code of life, actually in books that you can pick out and handle, open, and inside those books you actually see all these like letters. It's a code. It's a coded messages of our genes, our instructions to life, to how to make a human, basically. And this is all in those books. So these are arranged in chromosomes. So you can see the book for chromosome 1 is quite a lot. Chromosome 2, there's really thick books as well. And all you can see in there is the sequence. We managed to sequence the human genome, and that was the most amazing achievement. I mean, for me as a geneticist, of course. But obviously, we had to decipher that code. It was literally like a code that that they used in the war, for example, to decipher the messages that we're sending across the different adversaries. So when we are in the process of that. We're doing a really good job actually deciphering this code. But basically, we had to make sense of all these letters that were in our DNA. So why are we so unique? Why not everybody, not all of us have the same code? Essentially, if you think about you, this is you. We all started from one cell. The instructions from one cell. It was 50% from our father, 50% from our mother. But this one cell, all the genetic instructions actually was a combination of a series of combinations of new genomes that came back from our ancestors. So basically, we are a combination of everybody here. Random combination, random shuffling of our genomes, and then we become. This comes to culmination to us. So basically, as I said to my students, we are the survivors, basically, of what we, how our ancestors did. So they survived quite a lot of stuff, and we are the results. So the first 5 days of a human is actually here is one cell human. We have the zygote, which we call the pre-embryos, the pre-plantation embryos. And then they sort of start to divide, and then at day 5 we have an implantation. But by that time, we do have all the information that we need to create for an embryo to implant and continue on for a pregnancy. And again, when the two genomes fuse, create a new potential human. I say potential because a lot of the pregnancies in humans do not result in a live birth, okay? So that also is another human characteristic. And then how do we get there? How do we know what we have inside our genome? So we have the state of the art. So basically, this is the sequencing. So we started sequencing the genome, trying to take the information from biology to look at what letters we've got in those code. So this was the first sequencers in the Sanger Institute, and these are quite big. And back then in 2000, when the first Human Genome Project actually was announced that it was deciphered, not deciphered actually, sequenced, meaning that we just know the letter code. It took £2 billion to do, 10 years, and various labs across the world. But now, the present day, if you want to do your genome, to sequence your whole genome and find out what's in your DNA, you can pay from 200 or 500 pounds. You only need a few hours, and you use a couple of scientists can do that very easily. And you either use these machines or a little USB sequencer. So it's like a little machine here. If you compare this to this, you can actually see how far science has taken us. Where we are now in genetics is quite a rapid change. But it's catching up with us because it is very complex. We possess. Each human has a terabyte of data basically generated in a sequencing site. So a terabyte it's very, very difficult to decipher. It's a lot of information from each individual. And all of us have interactions with our environment. So each of us have small mutations, a series of combinations from our ancestors, but also we have different environments that we grow up. So our environment is shaping us as well. And it creates an effect of interaction. So it creates a lot of interactions between what we have inside us and what we exposed as an environmental factors. So we have different levels of environmental interactions with genetics. In cystic fibrosis, which is really a mutation that's happening, most of the elements would be genetic. So basically, because of the mutation, you'll have the disease. However, there's a small environmental chunk there. So, you know, depending on specific life factors or nutrition, could actually improve or make things worse. Adult onset diabetes is 50-50. So basically, you got the risk, you got the predisposition to have a diabetic, to have develop diabetes. But if you have sort of different lifestyles or different combinations of environmental exposures, then you might actually reverse the effects of diabetes or have a different interaction or different prognosis and development of the disease. And then we have HIV or AIDS. And then there is a big environmental component, of course, because it is a virus and it comes from the environment. But it also has a small genetic component because some of us are actually resistant to HIV infections. I deal a lot with fertility and infertility. So genetics and how it affects fertility in general, and especially males are affected by the environment. So, you know, they might be predisposed to certain risk of being infectile, but males really have they're impacted quite hugely by environmental factors like smoking, like toxins, radiation, that sort of thing. While females, their fertility is affected when they're still in their mother's womb. So you have a transgenerational effect there. So whatever the female's mom was exposed to will affect the female's fertility, basically. But in the males, it would be different. It would be throughout the male's life. So you have a lot of different things to think about in terms of fertility. And then for me, for example, this is what interests me. This is what my area of expertise. So you are one of a kind. You've got genetic variation, and that's there within all of us because we come from so many different people. And you also have a different environment that people are exposed to. So what we call, we're all mosaics. So, you know, you'll have different levels of mosaicism for everybody. So these colorations would be the maternal and paternal genomes. So all of us will have parts of our body that's more like our dad, parts of our body that's more like our mom, for example. We might look like one of our parents externally, for example, but internally we'll have our other parents sort of more so characteristic. So there is a little bit of expression, and this is, you know, how our phenotype, how we express our genes is actually the very, very interesting thing that's happening now. So we are all mosaics, we are all unique, and we apply this now. We started to apply this big level of complexity to real life problems. So we use genetics for a lot of different things. Nowadays, we use clinical diagnosis of genetic diseases like cystic fibrosis, like Huntington's disease. But also, as Nathan has pointed out at some point in his slides, we use it for prognosis and treatment options. So basically, we genotype cancers, or we look at specific mutations, and we understand how people respond to drugs, because, you know, we know that genetics is about slow metabolizers and fast metabolizers of drugs. So this information comes from genetics, and we have fertility treatments as well. Or you know, different types of diagnosis for prenatal diagnosis. I'm sure you've all heard of it. pre-implantation genetic diagnosis, which we look at embryos before they go to the mama's womb, and infertility investigations like sperm parameters, like endometriosis and things like that. So, and also we have commercial applications. So for fertility and infertility, which is what I do mostly, and I'll give you an example in a minute. A lot of things can go wrong, basically, but also there's one phenotype, infertility. So we have to go to do a lot of different types of investigations, presenting all these different levels of development to actually understand where that infertility comes from. So you have a very complex disease, but through biomedical science and through different investigations, you actually try to rule out certain things which are normal, and then looking at the abnormal side. So it could be from any parent, it could be from the implantation or pre-implantation. So there's lots of different types of things. So a couple of interesting cases. This is from my PhD, and this was when I was working in Great Ormond Street. So there was a young couple that was experiencing quite a lot of miscarriages, and they were very young. They were sort of mid-20s, which usually, the fertility-wise, they shouldn't be having a lot of problems. But they were healthy otherwise, but they had more than five actually miscarriages in the first trimester. So when they asked for some advice from us, we found out through a series of genetic tests that some of Nathan has described. We found that they had a translocation. So it was like in their DNA, some chromosomes were swapped letters, swapped chunks. So here you see there's normal supposed to be, and then this change, swapping of chunks of chromosomes together. They were probably the health wasn't adversely affected, but what was adversely affected was their fertility. So this was the reason for their miscarriage. The actual when we did genetic testing on their embryos, and we selected for the normal one, they actually had a successful pregnancy after the first try. So we don't know, but not all of us would know, because not all of us would have that sort of phenotype, but 1 in 500 of us will carry some kind of structural variation. So it's part of our uniqueness and how we predict the risk. And quickly, for the last sort of few slides, this is how we use now personalized information of health data in biomedical science. So basically, for coronary artery disease, which is a very can be very severe, is a type of heart disease and it's a leading cause of death in many countries. Nowadays, we do what we call risk profiling. So we take the genetic information, so we might do some risk profiling genetically. We'll take the lifestyle factors of the individual, and then we are trying to combine these two via certain risk profiles that we have to see where that individual fits. And we do a more personalized risk analysis of how to understand the disease as developing and what sort of risk can we assign to that particular patient. So for each patient, this will be different, but we have to take that all into consideration. So this is just an example for the heart disease, but nowadays, a lot of the more complex diseases like diabetes and other diseases are actually managed with risk profiling like that. So a little bit of a question now. So we have DNA now databases, one run by the NHS, the Genomics England one, which is a 100,000 genomes project. And then we have the Biobank, the UK Biobank, which is again another big database. What they collect basically is a whole genome. So volunteers give their blood and they get sequenced, the blood gets sequenced, the DNA. And then you have also all their medical records, the data, everything is in there. So we try to connect the dots basically. I don't know how many would provide a DNA. So if you if you can show me your hands, how many of you would like to provide a DNA sample for this type, or if you have, okay. Okay, so half of you, more or less, quite a lot of you actually, which is good. Of course, it is tricky. There's a lot of legal obligations that these, for example, in Genomics England, the 100,000 Genomes Project, they don't allow researchers to take any data out. You have to analyze it on that environment. So basically, you can't share a person's genome. Some other the databases, though, are fully open. And this will take me to the next question. And the last thing that we are going to look is direct to consuming genetic testing. I'm sure a lot of us have actually done some kind of genetic testing directly, maybe Ancestry, maybe just doing the whole genome or doing some mutations. You basically the companies would provide you with risks, bypassing the professional biomedical scientist or geneticist, and giving you a risk, for example, for coronary artery disease, traits, a lot of different types of health conditions, nutrition, maybe information athletics. So these companies will store your DNA there. You might be able to say, I don't want it to be public, but there are quite a lot of agencies that can tap into that, like the police and everybody. So basically, you do get people's genome out there in the open. You do get the genetic information. It is medical data. It's very complicated data, but people can actually tap into that. And there are instances that police have actually found relatives of the perpetrators like that, and they traced the through. I think in that case was 23andMe. So they can find your cousins and your brothers or anybody from your relatives that actually are related to you, and they can trace them back. So the question is, you have all this data coming back to you with no professional analysis, no actual advice, but how many of us do we really want to know all this information? I think that's what we have to think about. Is it something that we really need to know? Do we have to know everything about the DNA? What risks we carry? And how would we live our lives if we have all these risks, for example, coming at us without any explanation, without any cause? I think for me as a geneticist, I would like to know, because this is what that's my job. But I think it is creating, to people who not genetics, who are not, specialists, it does create a lot of confusion and a lot of different information that need to be deciphered before we get any conclusions. So in general, though, there is a lot of things happening in genetics, and I think that was the final part that I want to share with you. There's a lot of groundbreaking research, a lot of a lot of state-of-the-art that's improving. Things are moving towards a personalized environment. So people will go into any of the pathology hubs and get a more personalized approach to their treatment, to their diagnosis, to their prognosis. And a lot of the tests will actually become a bit more personalized. So this is what all these databases are for. My little bit of adding to this progress is providing specialist education and training, and hopefully with a lot of collaboration with USD, we'll have a bit more to do with that. So I would like to thank you. Please feel free to ask me any questions, or Nathan as well. But thank you very much for listening. Is it? Any questions, guys?
Weeks down to hours, and what's the next step?
This one. The next steps is just a continuation of what Anna said. It's going to be a lot more personalised. It's going to be a lot more genetic testing. One of the next steps that I've seen, which is just being started over in a hospital in London, St Thomas's, guys in St Thomas's, is particularly with microbiology, is well, I know I said before about trying to identify what the organism is. Bacteria takes time to grow, but we're using DNA technology. We can have a look at the whole. We can look at the whole genome and the whole population genetics of that particular sample and find out what is actually causing the infection right then and there. So that will take a couple of hours rather than a couple of days. And so through genetics and through a lot of DNA testing and a lot through technology that's coming through, it's going to get quicker, and we're going to get very personalized healthcare, but it's also going to be quite targeted as well, so that we can exactly find out exactly what's wrong. And then the next thing is, okay, let's develop treatments to be able to carry that on as well. That answer my question? Okay. Any other questions? Go ahead.
No, it's the same. So so you asked me if it's the samples of blood or saliva are more reliable. They're both the same. Usually we use blood because it does give a lot of higher quality DNA. But some of the saliva samples, they do pass a sort of quality assurance when they reach the labs, and then we only sequence them. So basically they need to be sequenced. So nutritional genetics also are very hit at the moment, very high in the agenda, but there's a lot of complications with nutritional genetics because they are so much affected by the environment as well. So you get a result, for example, about your nutrition, but then it all depends, like 50% of it, in my opinion, depends on what you're actually doing with your nutrition coming from the environment. So you might have a lactose intolerance, but because you're not eating a lot of lactose, you don't actually affect it in your life, or you might have other genes that counteract that intolerance. So we're still building a complex picture of what, especially with nutritional genetics, is still very complex picture of how we can use that information. So basically, how do we use it genetically? Genetic information is the next step. I think that's going back to Nathan, to Nathaniel. It is how we manage to decipher, not just decipher, but how do we make the code make sense for us individually and how we live our lives. The complication with toxins as well in the environment also comes into place now. So it is a bit of a conundrum for scientists. We have to bear a little bit, give some patience, a bit of patience with everything.
Yeah, absolutely. So you asked that, would the patient preference, or would they be sensitive to a particular antibiotic? Absolutely. And so generally that's through the patient notes. We liaise with the patient, and through chatting with them, with the consultants, would say to them, are you allergic to anything? If anything. We also monitor it quite closely as well. And so we take subsequent samples also just to see if it is clearing. Are they getting better as well? If they're not getting better, then we can change the antibiotics as well. So we don't give them, say, okay, just try this one, but we give in the lab, we give the consultants and the clinicians a wide variety of different antibiotics that they can try, because they might be intolerant to some of them. Hannah. The balance of the manual to automated techniques, it depends on which lab you're in, really. So some of the labs are very, very heavily automated. So biochemistry, hematology are very heavily automated. We have the big long tracks that I showed you a picture of. We've got some new pre-analytical equipment come in. So all you have to do is just basically put the sample on, press the button, and it will take it off and do everything to it. Whereas in microbiology, it's very hands-on. You still need to get the sample, you need to spread it onto agar plates. It is a very manual process. There are opportunities to have that automated, but that's not in the hub. So yes and no. Back. So there are quite a few different blood types. So we have there's A, B, AB, and O. And so these are predicated on whether there's certain proteins on the blood. And so, but then you've also got another factor on there as well called rhesus factor. So you can either be rhesus positive or rhesus negative. So there's quite a few different types that you can have. All blood types are pretty much the same. They all do the same thing. But when you, according to the different proteins that you have on the on your blood cells, if you transfuse that into someone else, you can have an immune reaction. Your body will actually kind of just fight against it because it's seen as a foreign component. As this is why we need to make sure that the blood types are exactly the same. There are some exceptions. For example, O negative blood is called the universal donor. So you can give that to absolutely everybody and no one will have a reaction for that. There's also a universal recipient. So someone with AB blood can have any blood type because they have A and they have B. O means they have no proteins on the blood, so they can give to everybody. Does that make sense? Yes, it's all passed down through genetics from parents to children. So with my genetics, so I'm A negative, but my mother is AB. My father is O negative. Generally, most of the population are O negative. And is through the genetics that you have will determine your blood type. So my wife is B negative. No, she's B. Yeah, she's B negative. B positive. Sorry, I'm A negative. And so my children can either be A, B, or O because of how the genetics actually work.
Certain countries will have different, so you will have rarer types of things depending on the country you're from. I think B negative is quite rare in some countries, while it's a bit more. So it depends really on the founder populations that were in these countries in the first place and what blood type they have. So obviously with new influxes you get better mix. So the more change you've got, the better, I think, in terms of genetics, basically. It is one person sequence, yes. The first person who to be sequenced is a male. It's a person who volunteered to have his first sequence, yes. No. Well, we are different. We are the same but different. So we differ in 1 in 1,000 bases of our genomes between each other. So between me and you, for example, we might be different in 250,000 places, and that will confer different changes to how we respond to drugs. Just yes, in the whole genome. Yes. So we might have the same gene. Well, I think, unrelated individuals can be up to, 300,000 differences. And that's small differences, but one small difference can actually make a really big change in the protein. And then that protein will act differently. So although we do have the same genes, so basically we have the same proteins, but these proteins can actually be slightly different between one another. So that's why we have all these little differences that together make a completely different human. So fingerprints, for example, how do police identify people? Is because of these differences, they compare people to each other. So yeah, the lucky person was one male who we are now referring to him as the reference genome, but we have sequenced quite a lot of people from that, yeah.
Thank you very much, although I expect people would be happy with me not having the microphone. Anyway, I found it absolutely fascinating. I'm a lay person, but I think I understand a little bit better now about these two fascinating subjects. One of the themes is about speed, how things have increased in their ability to get results, which of course is proving wonderful for populations of the world. And long may it continue. And I think there's probably many members of my family who are very, very pleased that there's only one of me. So if I can ask you all to show your appreciation for our two lovely speakers this evening. Thank you.